Thanks everybody for joining us at our next USDA ARS WSSA webinar series. We're going to let the room fill here for just a minute or so, and then we'll get everything started. Thanks for joining us today, everybody. I'd like to introduce Dr. Lee Van Wyken, Executive Director of Science Policy for the Weed Science Society of America, to kick things off.
Good afternoon, everyone, to this wonderful spring day here in Washington, D.C. My name is Lee Van Wyken. I'm the Executive Director of Science Policy for the National and Regional Weed Science Societies. This is the 6th in a series of 10 webinars, so we're just over halfway between the Weed Science Society of America and the USDA ARS lab here in Beltsville. And we're grateful to have Dr. Steve Young serve as moderator for today's webinar. Steve is our national program leader for weeds and invasive pests, and he's been in a job now for just over a year or two, and has a big job ahead of him. He's trying to attack some of our biggest weed problems, which has gone unchecked for a while here. Like behind me, I have hydrilla, biggest, baddest weed in aquatic systems. One of 112 weeds on the federal noxious weed list, and by far probably the most widespread federal noxious weed. So we need help in all areas and all forms: terrestrial, aquatic, natural areas, agricultural. So Steve is going to lead us into the promised land, right, Steve? So Steve has a great background. He's from Washington State originally, did a BS with or MS with Don Morishita at University of Idaho, went to UC Davis, was there with Dr. Jody Tomaso, and has stints within at Nebraska and Utah State. He was also the Northeastern IPM Center Director at Cornell, and now he is our National Program Leader for Weeds and Invasive Pests. So Steve, thank you for organizing this excellent webinar series, and take it away.
Well, thank you, Lee. I appreciate that introduction, and thanks again to WSSA for hosting this webinar series. And so I think a lot of you have been with us for the past several weeks, and appreciate your attendance, and look forward to hearing today a little bit more about the topic of mechanisms. So we talked about the plant competition topic last week with Dr. Horvath. And today we're going to hear a little bit about the molecular basis for what do we do on invasive plants. And we're going to have Dr. Matt Tancos here talking to us. And Matt is originally from the from Northwest Indiana, and received his BS in biology, specializing in genetics and ecology from Ball State University. He received his PhD in plant pathology and plant microbiology from Cornell University in 2016. Matthew is then researched the bacterial plant pathogen select agent Rothabacter toxicus. I know I probably screwed that up, but Matt, you can correct me on my my poor Latin there. But anyway, worked on that while a USDA ARS postdoc researcher at his current location, the Foreign Disease Weed Science Research Unit there in Fort Detrick. His program is currently investigating the use of plant pathogens as biological control agents to manage invasive weeds in the U.S., characterizing pathogen emergence and evolution in our agroecosystems, and investigating novel molecular-based invasive weed management strategies. So really, I think you're going to hear some cutting edge information today, research, and this is probably going to spur a lot of questions. So as we've done in the past, please use the Q&A or use the chat box. Either one is okay to submit your questions. We will have some time for Q&A at the end, so look forward to that as well. But anyway, I want to turn it over to Matt, and let's hear what you have to say.
Yes, thank you, Steve. And you still see everything all right?
Yes.
All right. So yeah, thank you for that introduction, Steve. And again, I just want to thank the Weed Science Society of America and ARS and yourself for actually starting the symposium series to really highlight some of the fascinating weed science research that ARS is doing. And another thank you, Steve, for extending the invitation to myself to present some of the research that we're doing right now at the Foreign Disease Weed Science Research Unit located in Frederick, Maryland at Fort Detrick. So when I was first hired into this position, which is about 4 years ago, I really wanted to incorporate recent advancements in the field of molecular biology that could be used to augment microbial-based biological control agents in the field, with the ultimate hope that we could get them to increase their efficacy and reliability in the field environment for our stakeholders and the American public. So non-native invasive weeds directly impact our agricultural systems, as I don't think, as I would assume everyone is well aware of here. But invasive weeds cost the US about $25 billion in damages and losses, plus an additional about $10 billion in control costs every year. So invasive weeds compete for limited resources. They injure livestock either physically or chemically with toxins. And also they impact plant health and crop health for our cropping systems by potentially serving as reservoirs for plant pathogens and insects and other pests. And also the displace native and beneficial plant communities and choke our irrigation systems, especially out west when water can be a precious resource. So I included several images of grazing livestock, and that's because rangelands are some of the most difficult areas to manage these non-native invasive weeds, just due to the sheer size, terrain, topography, and the diversity of plant species that they maintain. And in fact, rangelands consist of about 770 million acres of land in the United States. So that's about 30% of land cover in the US. And invasive weeds cost land managers over $6 billion every year in damages and management costs. But invasive weeds don't only impact our agricultural systems, they also directly influence and impact our natural and urban systems as well. And especially our natural systems that are vital because they feed directly both back into our agricultural systems and our urban systems. And I just put the value priceless because really they are priceless for the benefit that they give all of us. Invasive weeds also impact and lower water quality and quantity. They impact our urban systems with recreational use and associated businesses, whether it's aquaculture, tourism, or recreation. And then another thing that we're continuing to see both in our natural and urban systems is the increase of wildfires and the roles that invasive weeds play in exacerbating these wildfires. And it seems like this is occurring earlier and having more severe fires, especially out west throughout the season. And as I mentioned earlier, so we know invasive weeds can impact crop health, but they can also directly impact and indirectly impact human health in a similar way by harboring insects that can vector and transmit human pathogens or animal pathogens just as easily. So the CDC currently recognizes about 18 tick-borne pathogens. And if we just focus on Lyme disease alone in the US, that has medical costs that exceed $1 billion every year. And similar to the wildfires that we see out west, we also see the increase of new diseases in the news and the expansion of ticks and mosquito populations and those pathogens that they can vector. So invasive weeds truly impact every facet of American life, and you don't need to be a grower to see that impact of invasive weeds, especially when they can impact our national security and vital precious resources. However, biological control can be a very cost-effective approach to combat invasive weeds over these large, inaccessible, or ecologically sensitive terrains. But we also have new challenges that are being thrown towards our direction, and that can include climate change. So not only is the plant range expanding for these invasive weeds and invading new areas and new ecosystems, but also the expansion of the associated microorganisms and pathogens, whether they can influence crop health or human and animal health. And with the introduction of new plant species and their associated insects or pathogens into these new ecosystems and terrains, you combine that with unpredictable weather patterns and climate change, and that really influences and impacts disease development. So not only is that going to influence managing these diseases in our crop settings, but also in trying to cause disease in what I'm doing, which is trying to cause disease with plant pathogens to control these non-native invasive weeds. So there's a lot of dynamics that are occurring in the abiotic and biotic factors, and climate change is just exacerbating that. And we'll touch upon this challenge a little later in the presentation. So here at the Foreign Disease Weed Science Research Unit, we're located in Frederick, Maryland, and we are present at Fort Detrick, which is an active military garrison. But our research mission is mitigating the spread and establishment of invasive pests that threaten the United States agriculture, national security, and natural resources. So our unit is really split up into two different research areas. So the majority of our scientists are investigating invasive plant pathogens. So they're studying phytopathogenic bacteria, viruses, fungi, oomycetes, and with the ultimate goal is order to mitigate the disease developing in American fields. And also a lot of these pathogens are foreign and emerging or are select agents listed as select agents by APHIS. So we really want to keep these organisms out of the United States at all costs, but we need to better understand them in order to mitigate those risks if they are introduced accidentally. And then on the other side, the other group that I'm leading is looking at invasive plants. So we're kind of doing the opposite of what my colleagues are. So we're both investigating plant pathogens, but I'm trying to encourage disease and optimize it in the field in the ultimate hopes of controlling these non-native invasive plant species. So we're investigating different invasive weed management strategies, disease development, optimization and modeling, and also pathogen evolution and emergence and the role invasive weeds play in this topic. So both of our research areas at our research unit are investigating plant pathogens, especially foreign plant pathogens. But we also share is our unit's unique biosafety level 3 plant pathogen containment facilities. So at Fort Detrick, we have a 10,000 square foot biosafety level 3 plant pathogen containment facility. And this is actually the largest BSL-3 containment facility in the US that's approved by APHIS for research on exotic plant pathogens. And the greenhouse space alone is about 7,500 square feet of greenhouse space to do a lot of these experiments. And our facility has research laboratories, greenhouses, growth chambers, dew chambers, and also a lot of instruments for molecular studies and basic questions, and all the way to applied research and translational research. The entire facility is under negative air pressure. We have HEPA air filtration. We have our own wastewater treatment facility on site, so all effluent waste that comes from this facility is essentially autoclaved. And then also any material that comes out is sterilized. Only authorized personnel are able to access the facility after you get the proper security clearances. And upon entering, we all put on clean scrubs to do the research inside. And then upon leaving, we leave our scrubs and everything inside and we shower out to ensure that we don't bring anything out with us. So in total, the USDA ARS actually maintains 4 BSL-3 research facilities in the US. So 3 of those 4 facilities focus entirely on animal diseases. And we are the only ARS facility that, BSL-3 facility that is really focusing on plant diseases and exotic plant pathogens. So, with the weed biological control group that I'm leading right now, we really kind of have our research split up into 3 different approaches or missions. So the first one, and I'll just give a quick overview of this, is our conventional or more classical biological control research. So we're doing exploration of foreign and endemic plant pathogens for our given target weeds. Then we evaluate those pathogens in our containment facilities and do host range studies to demonstrate that they can be very host specific and also efficacious and cause disease like we would anticipate, try to optimize disease development and potential releases. But after doing all of that work, you have to submit all of this to different regulatory agencies, especially APHIS and EPA, in order to get that federal approval and state approval to release these organisms into the wild for weed management. And so that's the more of the conventional classical approach that we're doing. We're also investigating more of the molecular approach. And this is more of the basic research. And we're looking at characterizing pathogen emergence and evolution in weed reservoirs and the associated risks that that brings to our cropping systems, especially if we have some pathogens that are closely related that can infect weeds and our high value agronomic crops. Also looking at microbiome and virome characterization of these invasive weed populations. And so we know these invasive weeds are foreign coming into the US, but what else are they bringing in? So what are their microbes that they are bringing over, unique viruses that they're bringing over? What is coming over and what role will those potentially have in influencing our agro ecosystems and cropping systems in North America? Also looking at disease modeling and diagnostics, especially rapid detection for our agents that are approved to be released. We want to monitor them and evaluate how efficacious they are when they are released into the field. And the last approach and topic that we work on is more of the technology-driven approach. So how can we use advancements in molecular biology to augment these microbial-based biological control agents? So I'm not going to talk about the first two research approaches that we're doing at our research unit. However, if you have questions or interested in any of that, please feel free to ask a question or reach out. But for today, I'm really focusing on the technology-driven aspect. Again, what molecular tools can we use in biological control of non-native invasive weeds? And I want to highlight collaboration is key, because this is an ARS symposium highlighting the research that ARS scientists do. But as Steve Duke mentioned at the first presentation, collaboration is key. And I think we all know that. And it's really vital for some of these more cutting edge new approaches and technologies. You really have to have that expertise from a variety of individuals in order to see if these approaches are viable, feasible, and safe to ultimately use. So I list my collaborators on the slides as we go forward, but just have to remember their important contributions that we all work with each other. So biological control, the ultimate goal is to weaken or kill plants and ultimately suppress that weed population so it doesn't continue to spread as easily and really wreak havoc on our agro ecosystems. And this can be achieved either with plant pathogens or insects. And I know Melissa gave a talk a couple weeks ago looking at insects and the work that the ARS group in Florida performs trying to evaluate insects to manage these invasive weeds in North America. However, a downside of biological control is that it's not always 100% effective. It's not a synthetic chemical. We're dealing with a living organism that has its own requirements, its own conditions that must be met for it to survive, let alone do what you anticipate it to do or want it to do. So there is those challenges with biological control. However, the goal is, well, can we use novel molecular-based technologies to kind of augment this and really bring biological control into the 21st century and try to expand our capabilities for weed management. So today's talk, sorry about that, is really going to focus on 3 different projects that we're currently investigating. So the first one's looking at nano encapsulation. And then we're also looking at RNA interference technology, how that could be utilized and exploited. and then also genetic biocontrol or gene drives. So I'm going to discuss all of these today, some of the preliminary work that we're doing. We are still at the early stages of this, but hopefully give you a better idea of what the projects are, the research that ARS is trying to do, and essentially evaluate whether these are feasible tools and practical, and hopefully be able to utilize them in the future. And some of this research, which is high risk, high impact, is exactly what ARS was designed to do, is to really target these difficult questions with our extended 5-year project plans to kind of target these questions that are risky and require a lot of that upfront research to see whether they're viable and can be a practical tool. So first I want to discuss nano encapsulation technology and really the removal of environmental stressors that can really hinder biological control agents in the field. So in order to get disease, we need to have 3 components, and that's an aggressive pathogen, you need to have a susceptible host, and you have to have a conducive environment for disease to occur. And we refer to this as the disease triangle. So we can screen for pathogens, identify them, whether they're bacterial, viral, fungal, oomycetes, whatever it may be. And you can screen those isolates and strains and try to select the most aggressive one that will work for your given target, invasive weed. We can also demonstrate in the lab if our hosts are susceptible, if the unique genotypes that we have in North America or the United States are susceptible to the given pathogen that we're investigating. However, the one thing we cannot control is the environment. Environmental conditions, whether humidity, light intensity, water availability, temperature, UV pressures, these are things that are out of our control once it leaves a greenhouse or a growth chamber. So in our containment facilities, we can try to optimize disease development and provide everything necessary to really get disease. But once this is in the field, a lot of that is out of our hands. And this can be a big reason why individuals may think biopesticides or biological control agents are unpredictable or unreliable because they might not do the exact same thing that they do in a greenhouse setting or a growth chamber. But we want to be able to see if we can remove those stressors and make it more reliable for our stakeholders. So we hope to accomplish this with nanotechnology and nano encapsulation, and having the ability to really add properties, value to our products in the field. So nano encapsulation is commonly employed throughout the food and pharmaceutical industries, and they really utilize it to stabilize, immobilize, or provide the controlled release of an encapsulated agent, whatever that may be. But with new technological advances, lower manufacturing costs, it really makes this an opportune time to investigate novel micro and nano encapsulation technologies that could be utilized to improve the efficacy and reliability of these biological control agents. And so I'm actually collaborating with a scientist. He's a food scientist and an encapsulation specialist at Cornell University, Dr. Ali Abbasparad. And we're working together to try to see if we can do this with plant pathogens to target these invasive weeds. So I wanted to include some videos that he has from his lab, and it really just shows the technology and what you can do with nano encapsulation. So these are just dyes just to show what can happen. So the first video, hopefully it's all working. You can have a stable agent that is going to remain in its matrix until a given stimulus is added. And then you can have that slow release of your cargo into the environment to achieve your ultimate goal. In the subsequent video, this is different thicknesses and composition of your matrices, which ultimately influence the rates at which the agent is released. So the green is degraded first, and you have that release, and then you can have a delayed slow release of the red dye. So after seeing this, well, why can't we use this with our microbial biological control agents and actually having the ability to have them in a safe environment and have them foliar applied to the leaf surface, but they're not triggered and will release until the environment is conducive to disease. And also you could have a delayed response. So only some of it is released in a given time, and then sequentially more is released over time. So you're constantly exposing the plants to this inoculum. So that's the goal of what we're trying to do, and really utilize these chemistries to increase infection rates and also the survival of the organisms once they're in the field, and most importantly, field establishment. And this is really critical because a great thing about using plant pathogens to manage these invasive weeds is that you can get a first infection and then you can get secondary infections because, again, you're dealing with the living organism on its own. And in some organisms, they can persist on the dead plant material. And in subsequent seasons, you can have that reestablishment and further spread. So ensuring that you have a high amount of inoculum that very first application is really critical to get that establishment in the field. Oh, and I just wanted to highlight, so there has been research in this looking at more macro and micro-based products to pretty much increase the shelf life or the ease of delivery for certain bioherbicides. But what we're trying to do is add more value products to it, such as causing micro wounding or only being released under a given stimulus. So right now, with the collaborator, Ali, we're trying to do this with a model system to demonstrate that, yes, we can encapsulate through the different technologies an invasive weed pathogen and see if we can control and add different properties with chemistry to that to see if we can get a slow release with a specific stimulus or also cause micro abrasions or wounding to the plant surface to increase the likelihood of disease. And so we're doing this with a model bacterial system. We're playing with garlic mustard right now. And we're utilizing an endemic garlic mustard pathogen that I identified called Xanthomonas campestris pathovirine cannae. And the strain we're working with is very effective at garlic mustard. And it appears to have a very limited host range against this weed. But those host range studies are still occurring. But we kind of wanted to use a bacterial system as a model system because you can easily grow it and it's just easier to manipulate than a given fungus. But one of the things is with this bacterial pathogen is that you can really see nice disease if you have a wounding event occur. So these two images of a leaf is either the leaf was cut or it was pierced with a needle or wounded in some way. And you can have that rapid disease development. And this pathogen actually internalizes in the vascular system. So it can spread systemically throughout the plant and ultimately results in plant death. And another aspect is that not all pathogens are created equal. So what's cool about this technology is that we could tailor it to a given microbe and to a given host. So you can really work with each pathosystem and optimize it for that given environment and pathogen. So in this case, Xanthomonas requires wounding to really see that nice severe development and plant death on a very regular schedule. However, if you don't wound the plants, like what we would see in nature, if you just did a foliar application, you will still get infection as we see on the margins of the garlic mustard leaf. And that's because the bacterium is able to enter through the hydathodes on the leaf margins and access those vascular elements and spread throughout the plant. So you still get disease, you can get plant death, but it takes longer to get that desired effect of what we want. So how can we encourage that in the field? So the first thing is really, you have to know what the chemistry and the leaf surface is of your given plant. Obviously, this could be different for every weed that we're dealing with. But for garlic mustard, you have to see what kind of chemistries are going to interact with that leaf surface and adhere to it. So looking at the top surface and lower surface of the garlic mustard leaf, and then seeing again what chemistry is going to work best. So you can see these white droplets. So in no way is this nano or micro. So the bacteria is in there and it is encapsulated in those matrices. However, this is just droplets placed on the leaf. So you could visually see how well the substance would adhere or roll off the leaf. So this is not how it would look. You would spray it either in a mist or even in a powder form. You could apply it. So our first iteration of this, it didn't adhere nicely, and it would roll off the leaf, and any rain event would quickly wash it off the leaf. So change it up, change the chemistry, and then we're able to actually get tight adherence to the garlic mustard leaf. And again, these are just droplets of the encapsulated bacteria within it. But this shows that we can just change those chemistries and get it to attach to our given leaf tissue of choice. So ultimately, what we were able to demonstrate just at the early stages is that you can encapsulate, in this case, a bacterial pathogen with different steps. It survives that encapsulation process, it remains viable, and it still remains pathogenic. And we were actually surprised because this was just a quick encapsulation. There wasn't any additive properties incorporated to cause wounding to the leaf. But with our encapsulated bacteria, we were able to see those nice V-shaped lesions where it's accessing that vascular system of the garlic mustard. But in the non-encapsulated control, you really still see it near that leaf margin, and it's not really progressing as deeply. So we're excited about that. We're going to look at this further, add other elements to the matrices, but we also need to evaluate this under inhospitable environmental conditions, high light intensity, heat, temperature, water, low water availability, etc. So ultimately, what the potential is that we could, in a way, control these microenvironments, even if they're in harsh, unpredictable environments, which we can see more regularly with climate change and in disrupting that and influencing that. So the ultimate goal is that we can control all three aspects of the disease triangle in order to increase the likelihood we'll have nice field infection and ultimately pathogen establishment when it's released. So now I'm going to switch gears and move over to RNA interference technology and essentially hijacking the plant's defense system against itself. So RNA interference is an evolutionarily conserved defense response in plants and animals, and it's used in order to protect against viruses, viral intermediates, and transposable elements that can wreak havoc to genomes. So in summary, when double-stranded RNA enters the cell, it triggers this defense pathway that results in the double-stranded RNA being diced up, and those individual fragments are then used to identify specific sequences that share homology to that fragment that was cut up, that original double-stranded RNA. So this pathway is essentially using that RNA template as a guide to find any homologous regions or similar RNA, such as messenger RNA, and then degrade that so that product isn't there and you can inhibit that transposon or those viral intermediates. So RNA interference has been used thoroughly in molecular biology over the years, and it's great for investigating cellular pathways and gene functions. And so you can transiently silence a gene and its product and see what role it plays in the cell process. So it's a great tool to use in molecular biology. And more recently, the idea has, well, what about applying this as a management tool, especially topical applications, foliar applications of double-stranded RNA for disease suppression. And this has been demonstrated and validated several times, looking at protecting against plant viruses and aphid-transmitted viruses, and also pathogenic fungi. And so the question is, well, if we can use this to suppress diseases, well, why can't we use this against the plant itself to make itself weak or more susceptible to disease or less fit in a given trait? So there's numerous advantages to this approach. You can topically apply it. There is evidence that it can be absorbed through the leaf. Obviously, this depends on the plant and the leaf structure. There's a lot of challenges in that. But it's a non-transformative way. So you get transient gene silencing. You're not having a genetically engineered plant by any means. It can also be made species specific because again, we can target unique sequences that could be present in only one species. So it's very fine-tuned control mechanism. And it's comprised of inert nucleic acid. This cannot self-replicate. It's not a virus. It doesn't have that capability. It's just a double-stranded RNA or RNA fragment. It's environmentally sustainable and it naturally breaks down. So RNA isn't exactly that stable, especially in an environment with active nucleases, UV potential, etc. So that's a good thing and a bad thing. But with encapsulation and especially nanotechnology, we can overcome those limitations. And this kind of ties into the previous talk about using nanotechnology to encapsulate pathogens. Well, you could also do that to encapsulate double-stranded RNA and its products. So I just briefly wanted to highlight a recent study from 2017 in Nature that was able to really show how nanotechnology can increase the reliability of this double-stranded RNA and as a potential agricultural tool to manage, in this case, diseases of plants. So they were looking at a viral pathogen. Hopefully, I don't know if you can see my mouse, but with the first images, if we just look at this quickly, the water is the negative control and you'll see disease symptoms, the viral infection. The LDH are these clay nanosheets that the group designed. Again, just the clay nanosheets on their own will not prevent viral replication. Now, with your double-stranded RNA just being applied to the leaf surface and being absorbed by the leaf, they're able to see the suppression of that viral infection up to 5 days after co-inoculation. However, by 20 days, it's not playing any role anymore because it's washed off, it's degraded, and it's not going to have an impact. But their last column of images is their bioclay. So this is the double-stranded RNA that's encapsulated in these clay nanosheets. And what they were able to show is even after 20 days post applying this on the leaf surface, you can still have protection. You can still have that ability to suppress that that virus in the plants. So again, really just shows the potential in new technologies that can be used to overcome some previously considered limitations. So our approach is, well, why can't we use this, this defense pathway against the plant itself and really a genetically tailored weed management tool that could be tailored to any given plant species. And again, we can make it species specific. And I have to highlight our collaborator, another ARS scientist, Michelle Heck, who's at our Cornell or at our Ithaca, New York campus. She's also part of Cornell University and the Boyce Thompson Institute. So what we're trying to see is, well, what if we see the additive effects of using RNAi and augmenting that with a plant pathogen or a biological control agent? So if we silence a given gene using RNAi technology in a plant, you still have the plant remains maybe stunted, but still viable, still can reproduce. So you still have that. But on another plant, you can infect it with a virus. Maybe it's just a benign virus. You can still see symptoms, but you can still have a healthy plant that can reproduce and spread. But it's that additive effect of silencing a gene, transiently silencing it, and co-infecting it is where you can get plant death and potential control. So this can be used with either co-infecting with a biological control agent or just applying that double-stranded RNA and then letting opportunistic and endemic pathogens kind of jump onto this susceptible plant after it is being suppressed when we're targeting the given gene of choice. So we're currently at the early stages of this, but we're evaluating specificity, deliverability, and the efficacy in model plant systems, but we're also doing it in the invasive weed garlic mustard. So we're kind of doing it side by side with this. And we can grow up the double-stranded RNA in the lab, in vivo systems with E. coli. You can also synthetically produce it. But the ultimate goal is that, yeah, we can evaluate this, demonstrate that it can be species specific. Even if we're targeting a conserved gene that's highly conserved, we want to validate and prove that you can still make it very specific. And then also, is it going to work, and can we make it efficacious for deployment? So we'll talk some more about challenges towards the end of the presentation, but there are challenges to this type of research. So again, host specificity, and we hope to accommodate that with utilizing model plant systems, well-known genomic plants with well-known genomes, trying to target these conserved genes, but make it very targeting specific sequences to make it species specific. And then another challenge is weed genomics. As Steve Young mentioned during the first talk, there's not a lot of energy going into sequencing a lot of these invasive weed genomes. But with lowering costs, next generation sequencing, and also focus sequencing, we can overcome some of these limitations. Stability and delivery, as we saw, can be remedied with nanotechnology and nano encapsulation. And then the last one is regulatory. Again, we'll touch upon this at the end, but there's a little hope because in New Zealand in 2018, they determined that there'd be no federal regulation of exogenous double-stranded RNA. So in the remainder of the talk, I want to focus on our last topic, which is a gene drive, potentially used to disrupt plant reproduction and limit the population spread. So myself and collaborators at the University of Minnesota, so Doctors Alan Smith, Neil Anderson, and Roger Becker, we're working together to see if, in wanting to really evaluate the feasibility, viability, and safety of a gene drive to manage non-native invasive weeds. So we're at the early stages of this, but we're wanting to evaluate the invasive weed common Tanzi and kind of use that as a gene drive model system to, again, see whether this is a viable, safe, and feasible weed management tool. So common Tanzi is an invasive Eurasian perennial, an Asteraceae. It's considered a noxious weed in multiple states, toxic to livestock, and acts as a reservoir for plant viruses and other pathogens. However, currently there's very limited control options that are available to manage this weed. And that's because it has a widespread distribution. There's no approved biological control agents. I think there is some research going on in Europe with collaborators in North America trying to investigate. I think they have a handful, one or two insects that they're looking at. But the host specificity is looking like it's an issue because we have native, closely related Tanzi relatives. And some of those species are threatened or endangered. So that makes it even more challenging to get a biological control agent to be released for this invasive weed. But we think this can be a great model system because it's sexually reproducing, it's self-incompatible, which is important. It's a diploid, short life cycle, and it's a prolific seed producer, up to 50,000 seed per plant, and it's insect pollinated, which can really aid in the dispersal of a potential gene drive. So what is a gene drive? Essentially, gene drives are self-replicating genetic constructs that bias the inheritance of a given trait that we want to put into an invasive weed population to control it. So therefore, thereby we can result in population level control of a non-native invasive weed. But how does that work? So put together the schematic of just looking at the inheritance of a given gene. So say we introduce a modified gene that we wanted to put into the population to assist with management. However, we're really limited by Mendelian inheritance because once the modified plant breeds with wild type, the progeny only has a 50% chance of inheriting that gene. And as you move lower down, subsequent generations, you really can't flood the population with a given gene. That could result in population level control. However, with a gene drive, you can kind of force that trait, that gene or disruption in an entire population. And as you see, you can, in theory, get almost 100% inheritance of the modified product. And then you can have population level control. So there's a lot of benefits to a gene drive, is that, and I'll discuss this a little further, but this can be, again, very species specific. We can target a unique sequence that only a given species has, and in our case, common Tanzi, and even show that it's not going to inhibit or work with native relatives of Tanzi, such as those threatened or endangered species. So in theory, you should have minimal environmental or collateral damage. So what actually allows a gene drive to be functional today and potentially as a management tool is the advances in molecular biology and especially CRISPR gene editing technology. So CRISPR is essentially, just a quick, is a bacterial defense mechanism that's been utilized in genetic engineering. So we're exploiting what was a bacterial defense immune system and using it to our advantage to cut at specific sequences in a given genome. So with normal genetic modification, we can add our gene to the chromosome in a given site. However, this is limited to only 50% approximately of the population or the progeny inheriting it. So with the gene drive, you really have to have three components that are going to be inserted into this. And that's represented here. So we have Cas9, which is a CRISPR-associated nuclease. And this enzyme will actually cleave the DNA and make a double-stranded break. We have our gene or cargo, altered gene or cargo that you can introduce with the drive. And then you have your guide RNA. So this is a unique sequence that's going to be specific that you design to your given species that you're wanting to target, that gene sequence that you want to target and cut. So then it has that recognition site, it cuts, and that whole construct can then be transferred to the other chromosome, and you can essentially get homozygosity. So the gene drive is self-replicating because it's a way of copying and pasting this construct over. And just a little more background on this. So first you have that Cas nuclease is employed. It utilizes that guide RNA to find that exact sequence that needs to be cut. And once that nuclease cuts that sequence with the guide RNA, this really is threatening to the cell to have a DNA cut. So because your DNA is damaged. So the cell rushes to repair these breaks, and in the hopes that it will use this mechanism of homology-directed repair. And this allows, so the cell utilizes that other chromosome as a template to then fix that double-stranded break that you put in there with this technology. And in doing so, it uses that copy and paste mechanism. And then you ensure that this construct is on both chromosomes, and you can kind of force it into the population in that way. So how is this going to work as a management tool? So our goal is to target a female fertility gene. So in this sense, we'll limit seed production and viability of the seed, but we're not going to impact plant growth or fitness in another way, because you can still have viable pollen that can potentially continue the gene drive and force it into the population. So by targeting that female specific gene, fertility gene, so we won't have seed, but we can continue that gene drive with the pollen. And that's demonstrated here in the little schematic I drew up. So the red is the pollen that can be further spread, fertilized, and then again, you can spread it further in progeny. And this could ultimately result in population level control. And again, since Tanzi utilizes pollinators, this too can be a great way to really ensure that it spreads in a given population with the ultimate hopes of managing it, utilizing this technology that can be very specific and safe to control. So kind of just to wrap things up, so there's numerous challenges to the projects that I was discussing. Again, we're at the early stages of all of this, But it really highlights, and what I think is important, is that we constantly have to be innovating and diversifying our research in order to identify new tools that could be used in our arsenal to control these non-native invasive weeds, especially with the advancements in molecular biology. And this is exactly the type of research ARS was designed for. It's high risk, high impact projects, but they're essential to see whether a tool or technology is going to be feasible, viable, and most importantly, safe to actually be utilized in the field setting. But only through research can we mitigate these risks, work around that, understand it, thoroughly design it, and especially test this in a highly contained, controlled research facilities, such as what Fort Detrick possesses and the Foreign Disease Weed Science Research Unit. Now, I know a lot of questions will probably be about policy and regulatory and whether this will be accepted or adopted, whether RNAi or nanotechnology or gene drives. However, as a scientist, I have to highlight that ARS is a research institution. We're not a policy institution. That's not our job. Our job is to show that whether the science, again, is feasible, viable, and safe. And then that is given to other agencies, determine whether this can be approved at the regulatory level. And again, we have to also work with stakeholders and work with them and alleviate some of these concerns that might develop at the beginning, because we don't want to rush anything and deploy this in the field, and it's going to have downsides that might be overlooked. But again, numerous labs are looking at gene drives, if we just focus on that, whether in animal systems, and we're looking at it in a plant system. Another hurdle is weed genomics, like I previously mentioned. Really having to sequence more of these weed genomes to employ some of these new technologies. So we're currently sequencing the common Tanzi genome as we speak. So that will be a great benefit for the research moving forward. And then again, stakeholder engagement has got to be key, and really work with them throughout the entire process as we move forward in some of these new technologies. So with that, I will be happy to take any questions. I thank you all for your time. And I want to just thank all of my collaborators at Cornell, ARS, and the University of Minnesota for doing some of this great research. Again, we're at the early stages, but hopefully in a couple of years, if we do this again, we'll have some great progress about the technology and what's been happening on that front. So thank you for your time, and happy to take any questions.
Yeah, thanks, Matt. That was great. A lot of really good information there. And I think we'll just cut right to the Q&A box. If you can see, there's a question from Dustin. Maybe you could answer that one first. We'll kind of go in these in order as time permits, but
yeah,
maybe address that one there. And
yeah, so the first question is, I know one of the biggest negatives of encapsulation methods have a dramatic effect on pollinators. Does the nano encapsulation encounter the same issue? So this is still early to say whether it's going to influence pollinators of that, but the chemistry that we're using is safe for the food industry, and that's why we're working with the food scientists. So this is all safe organic material that's going to be incorporated into these matrices. Let me pull up the questions again. So no, we don't foresee that being an issue, but that is something that is going to be investigated as we move towards that. And I think another thing you can do is kind of control when you're going to apply your agent that's encapsulated, maybe not when it's flowering. And usually for a lot of these, you kind of want to target the plant, the invasive weed, at the earlier stages, so you can have a greater impact on its fitness. So you hit it when it's younger and have a greater chance of controlling that invasive weed.
Another one there from Leonardo. I don't know if that one's, it's a little lengthy, but maybe address that one.
Let me try that. So I understand, the question is, I understand that if the control agent is an active component from a pathogen, that it can, oh, hold on, getting stuff moved. Understand that control agent is an active component from a pathogen, then it can be controlled. But if the biological control agent is a microorganism itself, how can it be contained once it is actively reproducing in the host? wouldn't you be concerned about transmission to native plants, which might provide a suitable host or an unknown host in both an agricultural and ecological setting? So I'm assuming this goes, again, just general biological control or encapsulation. But no, that's part of this. So I didn't go into the regulatory process of just getting the approval of just a plant pathogen. That has to be approved by APHIS and also EPA. And we have to do extensive host range studies to demonstrate the specificity of these pathogens and to ensure that they are going to be specific to our given target. So a lot of research really goes into those host range studies. So that shouldn't be a factor, because we should know whether it's going to be host specific before it even happens.
Yeah, great. Question there on polyploid weeds, more likely to develop resistance to RNAi or genetic biocontrol than diploid weeds. And actually, that was one of my questions, Matt, with what you're doing on Tanzi, which seems like a model plant. Relating this to others that aren't specifically good models, is that going to work as well? Or maybe the challenges are going to be even more with those non-model plants?
Yeah, and it's a great question, because this really is going to be a challenging thing. And a gene drive might necessarily be the silver bullet for invasive weed management. It might only actually work for a handful of these invasive weeds, just because you need to have those specific requirements to make it feasible. So so a variety of things can influence that. But yes, whether it's a diploid, polyploid, that can be an issue, and that's something that we didn't want to try to focus on, and that's why it wouldn't be a model system in this case. But yeah, that's a lot of unknowns, and again, more science really has to address those questions.
Yeah, great. We've got a three-parter here. It's also in the chat from Olivier. He's in Berlin, so we've got a few questions about he's talking about soil. In your case, the agent will be applied to weed leaves. What about weed like Cuscuta species that are leafless? So plants with no leaves.
Yeah, it all depends on the pathogen that you're dealing with. So you could use soil-borne pathogens and could release that into the environment, into the soil nearby the plants. And we know this for several pathogens that they can infect through the root system. So yeah, it doesn't have to be a foliar application. It just can be applied in that sense. But again, it really depends on the pathogen and the pathosystem. But yeah, we don't want to limit it to just one type of organism.
Yeah. He's asking about, we'll just skip that. The gene drive technology is a bit of a concern.
So which one are you on now?
I'm on the third part of his question. So if the weeds are eliminated, won't the ecosystem be disturbed?
Well, in this, we're trying to remove these non-native invasive weeds because they already have disturbed our ecosystems. And again, with a variety of things they can harbor, either impacting crop or animal and human health. So no, it will be disturbed if we're able to remove them, but that would be the goal of what we're doing, is trying to remove these non-native invasive weeds from these new environments that they're colonizing.
Yeah, and I think that, I mean, I've seen papers on this topic in journals and from the policy, ethical kind of approach, as well as the main science, research aspects. So I think there's both of those issues, at play here. But Matt, like you said, this is ARS conducts research, and it's up to others to figure out how does this fit into our integrated weed management systems, if that's something that we should be doing. So
yeah,
one
go ahead.
Go.
I was just going to say, yeah, none of these approaches are going to be a standalone silver bullet to manage invasive weeds. They're all going to funnel back into that integrative pest management and utilizing different approaches, different tools, depending upon the invasive weed that you're dealing with.
Mm-hmm. Yeah, because we had that same discussion a couple of weeks ago. I think it was maybe with Melissa's talk, if I remember. weeds, so we're dealing with a biological entity here that constantly is, you know, changing. And so when we feel like we've got things figured out, all of a sudden it decides, well, that's not necessarily true. So I think you're right. It's just like, and we all know this, right? One tactic is not the answer. This is part of an integrated approach. And so I think that's really valuable. We got probably a minute left. There's any other question? I see one on exogenous application of DNA and what's the efficacy of the uptake of these dsDNA?
Okay, I see that one now. Yes, so this, again, this is at, with new articles coming out all the time, we're still at the early stages of this with actually being a viable tool to manage plant species. So a lot of the current work is looking at model systems, but it really depends on the structure of the leaf, the plant itself, how easily it is absorbed. Does it need a kind of wounding? Does it need different adjuvants to allow it to enter the leaf? So these are all active things that need to be addressed, especially given your target weed and what you want to focus on. So in the case with like Nicotiana or some of these model systems, you can have variability in how much is uptake, how much is taken up through the leaves, whether it's systemically moved throughout the plant, and how efficacious it is. But again, comparing Nicotiana to garlic mustard or a different invasive weed, it's all going to change, and that has to be played out and investigated and optimized.
Yeah, great. Well, thank you, Matt. Appreciate your this information sharing has really been great, and we're getting a lot of great comments about people wanting to see this again. So just a reminder, yes, this will be posted to the ARS landing page as well as the WSSA landing page. So feel free to go there and view that whenever it's convenient. It'll probably take a couple days before it gets there, so be patient. Anyway, thanks again, Matt. I want to just tell everyone that next week we're going to continue our last presentation on mechanisms, and we're going to talk about addressing herbicide resistance with alternative chemistries. So make sure you join us for that again next week, same time, same place, and we'll do this again. So appreciate everyone joining us, and have a great week, everyone. We'll see you soon.
Thanks everyone for attending.